In this lecture, we're going to learn a little bit about Mendel and Punnett squares. Gregor Mendel's the first person who accurately predicted how traits are passed from parents to their offspring, so he's considered the father of modern genetics. He happened to also be a monk from Austria, and he happened to study genetics because he did a lot of growing of plants, and specifically garden peas. He discovered several important things about genetics and how traits are passed, including the law of segregation, which states that allele pairs separate during gamete formation. So it means that if you have an allele pair, like lowercase t, when the cell that houses those divides, one of those is going to go to one cell and the other is going to go to the other cell. So those two letters separate from each other from a somatic cell to make two gametes. He also discovered the law of independent assortment, and he discovered this through dihybrid crosses. And what he learned is that when he's looking at two different traits in an organism, one does not influence the inheritance of the other. They happen to become inherited independently. So it means that like eye color and hair color are not inherited together. They can be inherited separately, so you could have a combination of traits inherited from your parents. He also discovered the rule of dominance. So when two different alleles are present, one is expressed, and that's the dominant trait, and the other is hidden, and that's the recessive trait. And that occurs when you have heterozygous allele pairs. Mendel discovered this because he grew a lot of plants, and specifically peas. So if you've ever eaten peas, they're delicious. Most of the time they're green, but he grew many, many plants, and he tested the crosses for lots of different traits for those plants. So he tested, which flower color is dominant, where flower position, seed color, seed shape, pod shape, etc. And so all of that data together, he was able to devise how traits are passed from parents to offspring. One of the ways that you can predict is a Punnett square. And so most Punnett squares, and the ones we're going to do today, are monohybrid crosses. So that's a cross that involves looking at two different parents, but only one trait from each parent. So looking at, for example, maybe flower color. So we have a pink parent and a white parent, and those two together, we can breed together to find out what the children or what the seeds are going to grow into when we plant the seeds. A true breeding plant only produces one type of offspring. So if you have a plant that is homozygous for a trait, it is also called a true breeding plant. A test cross is when you don't know what the plant is, and you can find out by breeding it and looking at the offspring. And the way you do that is to cross your unknown plant with a plant that's homozygous recessive, because that will allow you to see only which genes are coming from your unknown plant. Anytime you're breeding organisms, which people have done for many years for plants and animals to get traits that we want, you want to keep track of what generation the animal or plant is from. And so we do that by labeling the generations. So the P generation is the parent generation. The F1 generation stands for first filial generation, so that's the children of the P generation. The F2 generation is second filial generation, that's the grandchildren of the P generation. So that's how they're related. And the word filial is about son or daughter, so that's why we use that term. So here's an example of why you might breed two things together to find out what you get. So here we've got two pea plants. We have a green pod and a yellow pod. And when we breed them together, we get all green pods. And what that tells us is that this yellow color is most likely recessive. And we can bear that out with proof by making a Punnett square. So here I've taken the genotype of both, or of two different parents of a plant, and I'm going to generate the probability that they're going to have offspring of various colors. These two plants, I know their genotypes. So the top P here, this is big G, small g. That's the genotype. And big G stands for green pods, and little g stands for yellow pods. Then I have my second parent. That genotype is also capital G, small g. So both of those parents are what we call heterozygous for the trait. And then I can find out or predict what their offspring are going to look like by creating my Punnett square. So the Punnett square is just this box. It's got 4 little boxes inside of it. And I'm going to write the letters from the parents in the boxes to predict what the offspring are going to look like. So in my first box, I get 1G from the parent on the top of the box and 1G from the parent on the side of the box. And that's going to give me the genotype of the offspring. That particular offspring is going to be big G, and be a green pea. Then I can look at my next box. So here I've got small g, and I also have a big G, and that's going to give me the genotype big G, small g. That's heterozygous, which means it will also show the dominant trait, but it could pass the small g or the yellow trait to future offspring. In the next box, I'm going to take my capital G from the top box and drop it down. And then I'm going to take a small g from the side of the box and put it here. And so that gives me the genotype big G, small g, also heterozygous, so it's going to be green. And then in the last box, I have my small g from the top of the box and my small g from the side of the box, and that's going to give me small g, and a yellow pea pod. So that's how you do a Punnett square. You take the letters from the outside of the box, which are the parents, and then fill in the inside of the box to predict what the probability will be that the offspring will be a specific color or whatever trait you're looking for. So in this example, the probability that my plant is going to be green, for example, is 75%. The reason I know it's going to be green is because there's a capital G in the box. And so for green individuals, I have this one is big G, so that will be green. This one's big G, small g, so that will be green. And this one is big G, small g as well. It will also be green. So that's 3 out of 4, or 75%. I also get one plant, probably, that is yellow. And it's going to be small g, and the only way I can get yellow is if I have small g, so that's going to show up 1 in 4 times, which is the same thing as 25%. Those are the probabilities that I will get those different phenotypes. The same rules apply to other plants and animals. So for example, this is corn. Each seed on a plant or an ear of corn is pollinated by one of the hairs or the silks on the corn, and then it grows into a seed. So you can get corn with lots of different colors depending on the genotype of the species that fertilized it. So then yellow is actually recessive and purple is dominant, and you'll notice that this is definitely a plant that had purple and yellow parents, and so you can see both traits on this ear of corn. So now we can do an example Punnett square. In this example, purple corn is dominant to yellow corn, and we're going to use the letter R and lowercase r. So capital R stands for the purple allele, and lowercase r stands for the yellow allele. So we are going to make a Punnett square that shows us the F1 cross. So we're going to take these two parents, and both of these parents are capital R, small r, and we're going to make a Punnett square. So we're going to put capital R over one box and small r over one box. That's one of the parents. And then we're going to take this parent and we're going to put it on the side of the box, capital R, small r. Then we're going to fill in the box. So I have capital R from this parent and capital R from this parent. And then I have capital R from the parent on the side and small r from the parent on the top. And then I can put capital R here and small r here and small r here and here. And that's going to give me 3 out of 4 purple offspring and 1 out of 4 yellow offspring. And we can do this problem again with different parents if we want to. Let's do the same problem, but now we're going to use the parents from the pea generation. So we're going to have capital R on one side of the box, and we're going to have small r on the other side, and then we can fill in our boxes. So we have a capital R from this parent and a small r from this parent. Then we're going to do capital R, small r, small r, and capital R, small r. That gives all of our offspring the same genotype. They're all capital R, small r, which means we'll get 4 out of 4 that are purple and 0 out of 4 that are yellow.